Okay, so it's 9 o'clock sharp. So let's get started. So hello everyone. Welcome to our today's Zoom seminar. It's really my pleasure to welcome my friend and my colleague Dr. Hao Jin from NIH to present his recent work for us today. So Hao currently is NIH investigator in the laboratory of host immunity and microbiome, particular in the neuroimmune crosstalk unit. Hao actually got his undergraduate degree of science in the natural science from Shanghai Jiao Tong University, and then afterwards he pursued his PhD training in the National University of Singapore on the developmental biology of blood stem and immune cells. And then after he completed his training in Singapore, he then moved to United States, work as a postdoc in Columbia, where he switched to study the neurobiology of mammalian taste. And I think he was working together with Charles Zucker, which is quite a renowned neuroscientist in the taste biology field. And then after he completed his postdoc training, in 2022, he also decided to combine his PhD training and then postdoc training together to study the neuroimmune crosstalk. He actually started his independent research program as a tenure track investigator in the laboratory of host immunity and microbiome at NIAID, NIH. So Hao's lab particularly interested in leveraging emerging neuroscience and immunology tools to develop and apply integrative multimodal approaches to explore these physiological rich neuroimmune connections to review fundamental principles of neuron control of immune responses. Currently, his lab mainly focusing a few different areas, which including the role of body-brain axis in the control of innate immune responses, which I think we're going to hear today. And then second part is representation and regulation of distinct types of immune responses by the brain. The last part of his research area is to study the modulation of immune responses by sensory experience and the internal states. So I think without further ado, Hao, thank you very much for contributing your science with us, and then we are very much looking forward to your talk. Please go ahead.
Okay. Thank you, Chuan, for the introduction and for the invitation. I'm really pleased to share our work on this wonderful platform. So my lab is broadly interested in understanding how multidimensional neuroimmune interactions contribute to the modulation of immune responses and body physiology. We're fascinated by this diverse array of neural elements that pervasively innervate virtually all the barrier and immune tissues. And these neuronal elements are in close contact with immune cells and therefore constitute bidirectional neuroimmune communicative pathways that allow sensory signals to be sent up to the central nervous system and the motor commands to be sent back down to the periphery. And we're trying to figure out how this intricate neuroimmune network senses immune signals, reports immune information, and regulates both local and systemic immune responses. And today I'm going to tell you a story about how the vagal brainstem axis monitors and interregulates body inflammation and the peripheral innate immune response. As we all know, inflammation and the immune responses against pathogens and infections. Immune system and inflammatory response will be triggered to activate immune cells to promptly remove pathogens such as host disease, which helps the amazing healthy states. if the immune system is somehow compromised, such as in those immune deficient animals, few immune cells get activated. As a result, pathogens overgrow, often leading to the demise of the animals. However, the inflammatory response has this dark side. An out of control inflammatory response could produce autoreactive immune cells and mediators, which attack and destroy normal healthy tissue cells, leading to many debilitating consequences. So having a balanced immune response that is both efficient against pathogens and safe to the health is critically important for both the survival and fitness of the organism. In fact, many diseases and disorders arise or are aggravated because of imbalanced inflammatory or immune response. A weakened immune system could produce primary or secondary immune deficiency disorders. On the other hand, an overreactive immune system lead to many inflammatory diseases, including cytokine storms, many forms of autoimmune disorders, as well as allergies. And because of this importance of regulating inflammation, the organism evolves multiple strategies to keep this process in check. And within the immune system, the immune balance to get this right amount of inflammatory response is primarily regulated by a tug-of war between pro and anti-inflammatory immune cells and cytokines. For example, pro-inflammatory cytokines, TNF-alpha, IL-6, and IL-1 beta, which promotes inflammation, and IL-10, on the other hand, which antagonize pro-inflammatory cytokines to dampen inflammation. And the activity of immune cells is also heavily influenced by factors extrinsic to the immune system, which coexist with immune cells in virtually all the barrier and immune tissues. For example, commensal microbes have been shown to tune tissue immunity, and also pioneering work from Kevin Tracy and Isaac Chu's labs have shown that peripheral nerves, including the vagus nerve and dorsal ganglion neurons, play important roles in shaping immune responses in a number of tissue and infection settings. We instead ask the question of whether and how the brain might monitor and modulate body inflammation to ensure an adequate yet non-detrimental inflammatory response. The brain, as we know, is the central arbiter of body physiology, regulating many physiological processes from heartbeat, respiration to metabolism. However, how the brain controls tissue immunity remains largely unexplored. Anatomically, the brain is extensively connected with peripheral barrier and immune tissues by ascending and descending neural pathways. Sensory information from barrier and immune tissues can reach the brain by spinal or vagal sensory afferents.
So sorry, Hao. Can you hear me? Hao? I think the audio is somehow stopped. Can you try? We can't hear you. Can you say a few words? Yeah, I can't hear you.
From which side you cannot hear? So the anatomically, the brain is extensively connected with peripheral immune and barrier tissues. by ascending and descending neural pathways. Sensory signals from barrier tissues can reach the brain by spinal or vagal sensory afferents. And conversely, parasympathetic and sympathetic efferents carry motor commands back down to the periphery. So we wanted to know how this neuroimmune anatomic infrastructure allows the brain to monitor and intermodulate body inflammation and peripheral immune responses. And we learned that if the brain monitors and modulates peripheral immune responses, and the brain neuron should be activated by immune index and given peripherally. So we use a very simple immune stimulus, LPS, or lipopolysaccharide, which is a component of the outer membrane of gram-negative bacteria, to trigger strong innate immune response. LPS was given IP, and in search of active neurons, the brain was examined for the induction of FOS, a marker of neural activity. So we detected a strong bilateral labeling of CFOS in the caudal nucleus of the solitary tract, or CNST, in the brainstem, meaning that these neurons are strongly activated in response to LPS. We were also able to monitor the activity of these neurons in real time using photometry, in which we stick an optical fiber onto the CNST and then record calcium responses from these neurons that express genetically encoded calcium indicator, GCAMP. LPS triggers a slowly ramping response that peaks at about 2 hours after LPS stimulation. So LPS strongly activates CNS neurons. But how about other immune challenges? We found that a range of other immune stimuli, including LTA, flagellin, profilin, and xamolson, when they were given intraperitoneally, or strongly activate the CNST, suggesting that the CNST is indeed the brain center for detecting peripheral immune stimuli. To interrogate the function of these immune response neurons in the modulation of immune responses, we need genetic access to them. To gain genetic access, we turn to the TRAP system, a genetic trick originally developed by Li Chunluo's lab at Stanford, which could translate trend neural activity into the expression of crude kinase. So in essence, this system has a tamoxifen inducible crude kinase knocked into the C4 gene locus. So when there's sensory stimulation, in our case, the LPS stimulation, the C4 gene and hence the crude kinase will be expressed in responding neurons. In the presence of tamoxifen, the crude kinase will be activated to turn on the expression of effectors, but only in active neurons. With this genetic tool, we first validated to see if the TRAP system could effectively mark LPS activating neurons with a fluorescent reporter. What we did is to first trap LPS responsive neurons with a TD tomato reporter, and then perform the second cycle of LPS stimulation, followed by fast antibody staining. What we found is that the majority of the LPS trapped neurons and those labeled with TD tomato in red co-express FOS following the second cycle of LPS stimulation. So with this genetic tool, we could now manipulate the activity of these neurons and ask if they may have any roles in the modulation of inflammatory response. So we use the TRAP system to express the inhibitory drug receptor GI, a chemogenic silencer, which could effectively hyperpolarize neurons in response to a synthetic agonist CNO. What we actually did is to first inject AAVs carrying the crude-dependent GI receptor into the CNST of trap animals, and trap label those neurons with the GI receptor, stimulate those neurons with CNO to silence these neurons, and then finally examined what happened to LPS-induced inflammatory response while these neurons were chemogenically silenced. So this is a normal cytokine response to LPS in wild-type control animals. A single dose of LPS triggers a strong release of pro-inflammatory cytokines, including IL-6 and IL-1 beta, and as well as the anti-inflammatory cytokine IL-10. However, when these brainstem neurons were chemogenically silenced, there's a dramatic change in the inflammatory response. Pro-inflammatory cytokines overshoot, and concomitantly, the anti-inflammatory cytokine undershoot. So in essence, when these brainstem neurons were silenced, there's exacerbated inflammation in response to LPS. Then how about increasing the activity of these neurons? Again, we use the same trap system, but in this case, to express an excitatory drive receptor, GQ, in these neurons, and then test effects on cytokine responses while animals were challenged with LPS. And what we see is the exact opposite. And there's a dramatic suppression of pro-inflammatory cytokines, accompanied by a large increase in the anti-inflammatory cytokines. So essentially, when these a manipulation of these brainstem neurons could bidirectionally modulate the cause of LPS-induced inflammatory response. Silencing these neurons enhances inflammation, while activating these neurons and dampens inflammation. So next we ask, what is the genetic identity of these brainstem neurons, which have this remarkable capacity to regulate body inflammation? We first generated a single cell atlas of the entire CNS neurons, which comprise 20 different neural cell types. Then we sequenced individual LPS trap neurons, followed by aligning the trap-seq dataset to the reference atlas of CNS cell types. Using this approach, we found that LPS trap neurons are enriched in a subset of CNS neurons that express the gene dopamine beta hydroxylase, or DBH. We then engineered animals to selectively manipulate these DBH expression neurons. As you would predict, manipulation of these small genetically identifiable population of CNS neurons is sufficient to change the course of LPS-induced inflammatory response. And just like manipulation of trap neuron does, chemogenetic activation of DBH expressing neurons reduces inflammation, and pro-inflammatory cytokine goes down, and anti-inflammatory cytokine goes up. Conversely, anti-DBH saponin-mediated ablation of these neurons increase inflammation. So together, these results really identified these DBH expression neurons in the brainstem as the central immune regulator for body inflammation. And these neurons respond to peripheral inflammation, and then in turn provides a feedback modulation to ensure the inflammatory response to operate within the physiological relevant range. But what are the actual immune signals that activate these CNS neurons to drive the immunomodulation? And there are two candidates. One is the LPS itself, and the other is the cytokines produced during inflammation. And to tease apart these two possibilities, we took advantage of the NIAID mutants, in which the lack of this essential adaptive molecule blocks LPS signaling to produce cytokines. So in essence, these NIAID mutants are cytokine deficient mutants. We found that in the mid-80 mutants, LPS failed to activate CNS neurons, and demonstrating that it is cytokines rather than LPS itself mediates and activation of CNS neurons by LPS. But how do the cytokine signals get to the brain to activate the CNST? It turns out that the CNST is the primary target of the vagus nerve, one of the principal nerves that conveys sensory information from all the internal organs to the brain. If cytokine signals reach the brain via the vagus nerve, we predict that a transaction of the vagus nerve should block the activation of the CNST by LPS. And this is exactly what we see. Vagotomy significantly impairs the calcium response of these neurons to LPS, demonstrating that cytokine signals activate CNS neurons via the vagus nerve during LPS-induced inflammation. Since the vagus nerve is the route for peripheral immune signals to get to the brain, we reasoned that by monitoring how vagosensory neurons respond to cytokine signals, we should be able to infer how the brain is informed of ongoing inflammatory response. We implemented an in vivo calcium imaging platform to monitor vagal neuron activation. We use a VGLU2 driver to express the genetically encoded calcium indicator, GCAM, in all vagosensory neurons, and then image the nodose ganglia where the cell bodies of vagosensory neurons reside for their responses to cytokine stimulation. What we found is that cytokine signals activate a selective group of vagosensory neurons. But more importantly, pro and anti-inflammatory cytokines activate distinct and non-overlapping populations of vagosensory neurons. But who are the vagosensory neurons that respond to pro versus anti-inflammatory cytokines? We have a large collection of Cree drivers that mark different subsets of vagosensory neurons. We use these Cree drivers to target GCAM into each of these different subsets of vagosensory neurons, and then ask how they respond to pro versus anti-inflammatory cytokines. Using this approach, we found that TRPA1 expressing vagal neurons are selectively activated by anti-inflammatory cytokines, but not pro-inflammatory cytokines. Consistent with TRPA1 neurons responding to anti-inflammatory cytokines, if we drive the expression of the excitatory drug receptor GQ in these neurons and then activate them, we greatly suppress the level of circulating pro-inflammatory cytokines, and at the same time increase the level of anti-inflammatory cytokine. So TRPA1 expressing vagal neurons carry anti-inflammatory signals. But what are the vagal neurons that respond to pro-inflammatory cytokines? Following the similar approach, we found that KAKA expressing vagal neurons are selectively activated by pro-inflammatory cytokines, but not anti-inflammatory cytokines. And chemogenetic activation of KAKA neurons has a mild but significant effect on cytokine response. It moderately suppresses the level of pro-inflammatory cytokines without impacting the level of anti-inflammatory cytokines. So we uncovered two distinct vagal populations that carry distinct inflammatory signals. KAKA neurons detect pro-inflammatory cytokines, and TRPA1 neurons respond to anti-inflammatory signals. But do these two vagal immune monitoring lines send information to the DBH expression neurons in the brain to instruct immunomodulation? To answer this question, we performed rabies tracing experiments to map presynaptic inputs to these DBH expression CNS neurons. And the rabies virus will selectively infect DBH neurons and then jump retrogradely to mark their presynaptic partners. With this approach, we found that rabies virus carry a GFP reporter and jumping from the DBH neuron effectively label both TRPA1 and CALCA vagal neurons, and demonstrating that the DBH neurons, the central regulator of body inflammation in the brain, is synaptically connected with vagal lines that monitor cytokine levels. So far, I have described the ascending arm of body-brain circuit in which the pro and anti-inflammatory signals are detected by TRPA1 and CALCA vagal neurons, and then transmitted to DBH expressing neurons in the CNST to drive the immunomodulation that ensures an appropriate magnitude for peripheral inflammation. But how do these DBH expressing neurons regulate body inflammation that is far away from the brain? Most recently, we have been working to reveal the nature of the descending circuit that allows the DBH expression neurons in the brain to interface with the periphery. By tracing the projections of DBH neurons, we found that these neurons heavily innervate the retroventricular medulla, or RVLM, brain area that is also strongly activated in response to LPS. And the RVLM is the brain center that controls the brain's sympathetic outflow. And the RVLM contains descending neurons that project out of the brain and regulates peripheral sympathetic nerve activity by synapsing with sympathetic preganglionic neurons. We also performed the initial functional characterization of these RVM neurons. We found that chemogenic activation of these neurons, just like activation of DBH neurons in the CNST, reduces inflammation by inhibiting the release of pro-inflammatory cytokine IL-6 and IL-1 beta, and simultaneously increasing the level of anti-inflammatory cytokine IL-10. While more functional experiments are needed to cement the role of these RVM neurons, we believe that these neurons are part of the descending circuit that allows that could translate the activity in the ascending arm of the circuit into meaningful changes in the inflammatory response. Lastly, we ask whether exogenous activation of this anti-inflammatory circuit could have any therapeutic benefits. Overdose of LPS is known to trigger sepsis, multi-organ dysfunction, due to an out-of-control systemic inflammatory response. We were able to reproduce sepsis in mice by injecting animals with a lethal dose of LPS. All the mice receiving this lethal dose of LPS died by day 4. However, when this circuit was activated, either by activating TRPA1 expressing vagal neurons or DBH expressing brainstem neurons, most of the animals are protected from this sepsis-induced mortality. We then went on to see whether this restoration of immunobalance by the activation of this vagal brainstem circuit could go beyond LPS-induced inflammation. We landed on the mouse model of ulcerative colitis, in which mice drink from DSS-containing water and develop colon inflammation damage, resulting in a severely shortened colon length, heightened levels of pro-inflammatory cytokines, as well as the blood in the stool. Remarkably, we found that when we activate TRPA1 neurons in the vagal ganglia, multiple disease parameters are improved, including a much better growth morphology of colon, reduced levels of pro-inflammatory cytokines, as well as less fecal blood. So together, this data really highlights the potential of hijacking this circuit in the restoration of immune balance for inflammatory diseases. To wrap up, so our studies uses LPS-induced inflammation as a model to understand how the brain monitors and modulates body inflammation. We found that the brain monitors peripheral inflammation by exploiting the capacity of vagocentral neurons to sense cytokine signals. There are two immune monitoring lines in the vagal ganglia. The TRPA1 neurons detect anti-inflammatory signals, while KAKA neurons detect pro-inflammatory signals. And these two vagal lines then send information to the DBH expression neurons in the brain to inform the brain of the status of ongoing inflammatory response. These brain neurons, after receiving and integrating the information, triggers a top-down suppression of pro-inflammatory response and amplification of anti-inflammatory state, which is likely mediated by descending neurons in the RVIM via a sympathetic pathway. We believe this vagal brainstem body and brain circuit function as a rheostat to prevent a runaway inflammatory response, to ensure the inflammatory response to operate within a physiological relevant range. So this really adds the body-brain circuits as important components to the expanding list of factors involved in the multifaceted regulation of host immunity and immune responses. So with that, I would like to thank people from my lab and also our collaborators, especially Charles, my postdoc supervisor and mentor from Charles' lab. I initiated this project while I was a postdoc in Charles' lab, and also Georgia and Philip from Trans lab, who helped us with the DSS experiment and also the Seminole experiment. And I will stop here, and I'm happy to take questions.